Hello friends. Today's topic is micropipettes. Micropipettes play important role in measuring and dispensing volume accurately. Definition of micropipette is, micropipettes are precision instruments that are designed to accurately and precisely transfer volumes in the microliter range. Pipettes and micropipettes are used to measure and deliver accurate volumes of liquid. The difference between the two is that micropipettes measure a much smaller volume, starting at 1 microliter, while pipettes generally start at 1 milliliter. Use of micropipettes. Both pipettes and micropipettes are used in various laboratories, including 1. Microbiology. 2. Environmental sciences. 3. Medical. 4. Academic and research labs. Do you know how micropipette works? The tip is made up of plastic or glass is attached to the piston pipette. The tip is dipped into the liquid to be dispensed and volume is taken. The liquid volume to be dispensed is then expelled by sliding the piston between the volume defining limits. Do you know how micropipettes are designed? First design type is fixed volume. Second design type is variable volume. Micropipette are designed with respect to use. Let's see one by one. A. Fixed volume micropipettes. These pipettes are designed to dispense only its nominal volume, for example 100 microliter. We cannot measure more volume than 100 microliter. B. Variable volume micropipettes. These pipettes are designed to dispense volume selectable by the user within its specified useful volume range, for example between 10 microliter to 100 microliter. Types of micropipette. First is air displacement type A micropipette. Micropipette having air between the piston and the surface of the liquid is called as air displacement type A micropipette. Second is positive or direct displacement type D micropipette. Micropipette piston is in direct contact with the surface of the liquid is called as positive or direct displacement type D micropipette. Let's understand use of these pipette. First is air displacement type A micropipette. These are a type of micropipette, which are tools to handle volumes of liquid in the microliter scale. They are more commonly used in biology and biochemistry, and less commonly in chemistry because of the equipment is susceptible to damage from many organic solvents. positive or direct displacement type D micropipette. These are similar to air displacement pipettes, but are less commonly used and are used to avoid contamination and for volatile or viscous substances at small volumes, such as DNA. The major difference is that the disposable tip is a microsyringe, composed of a capillary and a piston which directly displaces the liquid. The plunger will stop at two different positions when it is depressed. The first of these stopping points is the point of initial resistance and is the level of depression that will result in the desired volume of solution being transferred. The second stopping point is, when the plunger is depressed, beyond the initial resistance until it is in contact with the body of the pipetteur. At this point, the plunger cannot be depressed further. This second stopping point is only used for the complete discharging of solutions from the plastic tip. Operating procedure. 1. Depress the thumb knob to the first stop. 2. Immerse the tip approximately 3 mm into the sample solution. 3. Slowly release the thumb knob to the initial position. 4. Solution is drawn up slowly into the tip. Do not release the plunger too quickly. Rapid release might draw bubbles in the solution and might splash solution on the non-sterile shaft. 5. Withdraw the tip from the sample solution. Place the tip against the side wall of the receiving container. 6. Smoothly depress the thumb knob to the first stop, pause, then depress the knob to the second stop. 7. Remove the tip from the receiving container, and return knob to the initial position. Do not let the knob snap back. Eight. Remove the disposable tip by firmly depressing the tip ejector knob. 9. Add as new tip and continue. Calibration of micropipette. Attach micropipette tips to micropipette. The desired volume is set and distilled water shall be aspirated. Dispensed into dry beaker and weighed. Repeat for 10 times. The mean of the weights are converted into microliter using the first formula and the percentage variation can be calculated by using second formula. Handling, care and maintenance. 1. Do not lay down the pipette when a filled pipette tip is attached. 2. Avoid difference in temperature between pipettes and pipette tips as well as the liquid, as this may lead to incorrect volumes being dispensed. 3. Do not allow any liquid to enter into the pipette. 4. Do not clean the pipette with acetone or aggressive solutions.